Welcome back to the next video. So this is the second part of the colligative property. In the previous video I have discussed about the colligative properties, and that is the part 1 of the colligative property. In that I have discussed about the relative lowering in vapor pressure, elevation in boiling point, and third is the depression in freezing point. Okay, so now the next colligative property is known as osmotic pressure. So when we discuss about the osmotic pressure, before we have to know what is osmosis. So let's start from osmosis. So what is osmosis? What is the definition of osmosis? Osmosis means the movement of solvent from lower concentration to higher concentration through semi-permeable membrane is known as osmosis. Clear? So once again I am explaining you about osmosis. What is osmosis? The movement of solvent from lower to higher concentration through semi-permeable membrane is known as osmosis. Now here the thing is that lower to higher concentration. Generally what you know, that what is the movement of the particles? Particles are always moving from higher potential to lower potential, higher to lower, but here I am writing it lower to higher concentration. Why I am writing it here lower concentration to higher concentration? Lower concentration means when the side in which the concentration of solute is low, that is called as the lower concentration. And what is higher concentration? Higher concentration means where more amount of solute is present, that is called as higher concentration. It means lower concentration means low concentration of solute but very high concentration of solvent. Lower for less concentration of solute. But when we will talk about higher concentration, means here solute and solvent both are present. Means solute concentration is more as compared to this solvent. So here the solute concentration is more, that's why this is called as higher concentration. If we will see in the diagram, there is a semi-permeable membrane. Clear? So this is the solution side, and this is the pure solvent side. So here there is only water molecules are there. Clear? But in this side water molecule is there, as well as there is presence of solute particles. So from the diagram you can say that here the concentration of the solute is minimum. That's why I have written here lower concentration. But if we will see the solution side, we can see that solute particles are more as compared to this side. That's why for this we are writing higher concentration. So lower concentration for less amount of solute, higher concentration for high amount of solute. But when we are writing lower concentration of solute means higher concentration of water, and if we are writing higher concentration of solute means lower concentration of water. So what happened in osmosis? The water molecule means the solvent molecules are moving through the semi-permeable membrane towards the solution. Clear? And this is called as osmosis. This process is called as osmosis. Now, how can we stop the process of osmosis? How can we stop the process of osmosis? So, osmosis can be stopped by applying pressure at the solution site. Means applying pressure in the solution site. In the solution site, what happened? We will apply a force, reverse force towards the movement of the solvent. Means from which direction the solvent is moving, from the opposite direction we are giving the pressure. So automatically what happened, at a particular temperature the flow of solvent from solvent to solution will stop. At a particular pressure the flow of solvent from solvent to solution stop, and that particular pressure is known as osmotic pressure. Clear? So what is osmotic pressure? Osmotic pressure is the minimum pressure that stops the flow of solvent through osmosis is known as osmotic pressure. The minimum pressure that stops the flow of solvent through osmosis, means through osmosis whatever the solvent is moving, that will stop, and that particular pressure is called as osmotic pressure. Okay, so write the definition of the osmotic pressure: the minimum pressure that stops the flow of solvent from solvent side to solution side, is known as osmotic pressure. So we can write it: particular pressure that stops the flow of solvent towards solution side is known as osmotic pressure. So this is the definition of osmotic pressure. Clear? So means when we are applying pressure, what happens? Slowly the flow of solvent will decrease, but a particular pressure will come at which there will be no more flow of the pressure, no more flow of the solvent. That particular pressure is known as osmotic pressure. Clear to all? Okay. So this is about osmosis and osmotic pressure. Now we will see the application of osmosis. What are the application of osmosis? Application of osmosis. So what are the application of osmosis? So number one, absorption of water from the soil by the plants, or we can say the movement of water molecule to the different parts of the plant. Movement of water molecule towards the different parts of the plant is one of the application of osmosis. Number two, sprinkling the water over fruits and vegetables, over fruits and vegetables, vegetables to make them fresh, to make them fresh. So this is the another application of osmosis. This is the another application of osmosis. Similarly, to make pickle, the unripe mangoes are dipped in salt solution, Why? To remove the water present in the unripe mango, to remove the what? To remove the water from the unripe mango. Okay, so these are few applications of osmosis. These are the few applications of osmosis. Next, we will discuss about the osmotic pressure. Osmotic pressure in details. Osmotic pressure. So here already I have told you about the definition of osmotic pressure. I have told you about the definition of osmotic pressure. Now we will see that how it is mathematically, what is osmotic pressure. So mathematically osmotic pressure is represented by this pi sign. Okay, this is the symbol of osmotic pressure. So osmotic pressure is directly proportional to concentration, molar concentration. Is directly proportional to molar concentration. Molar concentration means number of moles of solute per volume of the solution in liter. So the osmotic pressure depends upon the molar concentration of the solute present in the solution. The osmotic pressure is directly proportional to molar concentration of the solute present in the solution. So that's why we can write it as pi is proportional to C. So this can be written as pi equals to CRT. Osmotic pressure is equals to CRT. So what is here R? R is the gas constant. T is the temperature. Okay, so these two are the constant terms. The rate R, that is the gas constant, and T, that is the temperature. So gas constant, we know that it is 8.314 or it is 0.082. That is the value of R according to SI unit or according to CGS unit. So first of all we will discuss that what is C here. So C is the molar concentration of the solute. So molar concentration of the solute can be written as number of moles of solute per unit volume of the solution into RT. So here we can see the osmotic pressure is directly proportional to number of moles of solute. That is B, NB. So that's why this is colligative property. Clear? NB. What is NB here? Number of moles of solute. So number of moles of solute is directly proportional to what? Osmotic pressure. That's why this is called as the colligative property. Now NB can be written as pi equals to, NB can be written as what? WB by MB into V. MB is the molar mass of the solute, WB is the given mass of the solute into RT. So this is the complete equation for the osmotic pressure. Clear? So this is the mathematical derivation of osmotic pressure. Now we will see the types of solution related to osmotic pressure. What are the types of solution? Types of solution according to osmotic pressure. Osmotic pressure. Clear? So what are the different types of solution according to osmotic pressure? Number one is the isotonic solution. Isotonic solution is known as the first one is known as the isotonic solution. So what is isotonic solution? What do you mean by the isotonic solution? Whenever the saline, you heard about the saline composition? In the hospitals generally the saline is provided to the patient. So saline contain 0.9% sodium chloride mass by volume composition. Okay, 0.9% NaCl mass by volume percentage. So this is the composition of saline water. So what happened when the composition of is equals to 0.9%, then the osmotic pressure of the two solution remains same. So what happened, the osmotic pressure when the osmotic pressure is same, when the osmotic pressure of the two solution is same, then it is called as isotonic solution. Clear? When the osmotic pressure of the two solution is same, then that is called as the isotonic solution. Means the composition of the solution is equals to 0.9% mass by volume percentage. Okay, 0.9% mass by volume. Okay, so this is called as the isotonic solution. Second one is known as hypertonic solution. Okay, second one is called as the hypertonic solution. So what is hypertonic solution? Hypertonic solution, when the concentration will be greater than 0.9%. When the concentration of the solution is greater than 0.9% mass by volume, when the concentration is greater than 0.9% mass by volume, then that solution is hypertonic solution. That solution is called as the hypertonic solution. In this case, what happened? The concentration of the solute is more. Hypertonic means the concentration of the solution is more. So what happened in this case? The water from the cell is coming outside. Means from the cell the water is coming out. The water is coming out from the cell and it leads to shrinking of the cell. Means if the concentration of the solution is greater than 0.9%, means it is higher concentration solution. What happened? The water from the cell is moving outside the cell, and that leads to shrinking of cell. And that is called as hypertonic solution. So generally what happens, whenever there is bacterial infection, bacterial or microbial infection, what happened? We are using the hypertonic solution. Whenever there is bacterial or microbial infection, we are using hypertonic solution so that the water comes out from the cell and the bacteria and the infections or whatever the microbes are there, that will get dehydrated. And we can overcome the bacterial infection. bacterial infection. So hypertonic solution is useful for preventing from bacterial or microbial infection. By dehydrating the bacteria, means by dehydrating the cell, we can dehydrate the bacteria and we can overcome the problem of bacterial infection. Clear? So this is the use of hypertonic solution. Next one is known as hypotonic solution. Third one is known as, this is the second one and this is the third one, hypotonic solution. Hypotonic solution. So what is hypotonic solution? When the concentration of the solution is less than 0.9 percentage mass by volume, less than 0.9 percentage. When the concentration of the solution is less than 0.9 percentage, now what happened? The concentration of the solution present in the cell is more than the concentration of the solution. So this is in less concentration. Hypotonic means less concentration solution. So what happened? Now the water move inside the cell, towards the cell. It move inside the cell. Okay, in the hypotonic solution what happened? The water move inside the cell and that leads to swelling of cell. Swelling of cell means, and the cell will burst. So what happened? Whenever there is hypotonic solution, means the concentration is less than 0.9 percentage, the water is moving towards the cell, from outside to cell. The movement of solvent is from outside to cell and this will swell up the cell and the cell will burst, and this type of solution is called as hypotonic solution. This type of cell is called as hypotonic solution. Clear? So these are the three types of solution according to osmotic pressure. If the osmotic pressure of the two solution is same, that is 0.9% solution is there injected in our body, then that is equals to the composition of our cell. Okay, that concentration of the solution is equals to the composition present in our cell. So at that time the osmotic pressure is same, that's why there will be no, they will not flow inside or outside the cell. But if the concentration of the solution is greater than 0.9%, what happened? The water is moving outside from the cell. Okay, and this type of solution is called as the hypertonic solution. Hypertonic solution is mainly used for bacterial or microbial infection. Why? Because it dehydrates the cell and the bacteria. Due to the shrinking of the cell, the water comes out from the cell and dehydrate the bacteria and the microbes, and we can overcome the bacterial infection. Third type is the hypotonic solution. If the concentration is less than 0.9 percentage, what happened? The water move inside the cell and there will be the swelling of cell and the cell will burst. Okay, so these are the three types of solution. Next, last, that is related to osmosis, that is reverse osmosis. Reverse osmosis. Reverse osmosis. So now if we are applying pressure greater than osmotic pressure, if the pressure we are increasing at the solution side, slowly the movement of solvent stops. That particular pressure is known as osmotic pressure. Now if in the solution side, suppose this is the solution and this is the solvent and it is separated by a semi-permeable membrane. This is the solvent. What happened? Now we are increasing, means up to this pressure the solvent is not moving, means the flow of solvent is stopped. That particular pressure is the osmotic pressure. But if we will increase the pressure now, what happened? Now the solvent is moving from solution to solvent. Now the solvent is moving from solution to solvent. Okay, means now it is opposite. Okay, so now what happened? The water is moving from solution to solvent. Water is moving from solution to solvent. It means what? It is against the osmosis. It is against the osmosis, and this is called as reverse osmosis. This is called as reverse osmosis. Here the solute particles are not moving. Why? Because there is a semi-permeable membrane. But the solvent is moving from solution to solvent against the osmosis. That's why this is called as reverse osmosis. So what will be the definition? When the pressure is greater than osmotic pressure, the solvent moves from solution to solvent side is known as reverse osmosis. Clear? Is known as reverse osmosis. So this is about the reverse osmosis. And one thing, what is the application of reverse osmosis? That is application, we can see one of the application, desalination of seawater. Desalination of seawater means if we are taking salt water in the solution side, if we will apply greater pressure than the osmotic pressure, then the water is moving towards the solvent and the solute will precipitate. So reverse osmosis leads to precipitation of the solute. The reverse osmosis leads to precipitation of the solute, that is the separation of solute and solvent. Clear? Okay.